Hello, hello, testing, testing. Okay, so I think I will start in 1 minute. So firstly, I need to say sorry because I have some things to do tonight. I had something to do tonight lah, so I forced to earlier the class today. So actually, maybe got some confusion lah because I am very sorry because of my emergency issue for tonight. So I hope that everybody can join. Okay. So for today's class, actually, is about semester 3 lah. So for those who are taking exam semester 3, you can come, you can share to your friends also to attend this class. So actually today I will start in chapter 17. That is chapter 4 lah for this semester, right? For this semester. So this chapter, I don't know your school teacher already start or not. Uh, but whatever. So this chapter actually is very long chapter lah. It is the longest chapter in this semester and also most important chapter lah because it is very long. So actually today I will mainly focus on Mendelian inheritance. But if I have time, if I complete the Mendelian inheritance earlier, I will start to start the non-Mendelian inheritance one. So see the time lah. So if the time is enough, I will proceed to two topic lah. If not, I will only talk about Mendelian inheritance. So without wasting our time, so I will go to the Mendelian inheritance. So okay, okay. So I think Mendelian inheritance, everybody know about it, right? Because you are already learned in SPM. So I don't know you guys still remember your about SPM or not. But if you not remember, never mind. So this is just a recap lah, recap chapter. So actually, non-Mendelian inheritance, as we know that we will learn about monohybrid inheritance and also dihybrid inheritance, right? Law of segregation and law of independent assortment. I hope that you all still remember these two laws. Okay, so when we talk about the inheritance, so about the history of inheritance, who found the concept, the knowledge of inheritance? This scientist, Mendel. He is the biologist, the fathers of genetics. So it is the first scientist, first biologist, first people that discover and found the inheritance theory. So actually, how he found this theory? Actually, he did the many experiments using P plants. So when he used the P plus to use the P plus, he found that there are many interesting characteristics about the plants, about the when they cross together, and then the genotype, the offspring of the P plus are also different also. So it makes the as many experiments, thousand times of experiments using P plus to study the knowledge of inheritance until now be applied. Okay, so why he choose P plus in his experiment to discover inheritance? So actually got five main reasons. The first reason is P plus is easily to grow compared to other plants. Right. Because if you compare to other plants, like woody plant, because pea plants is non-woody plants. Normally, if you choose woody plants, the the plant is not easily to grow. And then got some type of plant they require very specific environment, specific condition to grow. But pea plants is easily to grow. If you got any minimum requirement, then the pea plants already can grow. And then B, short life cycle, it can easily grow, become adult stage compared to other plants, if compared to human also, because human require about 18 years, right? 18 years until require normally 18 years become adult. But plants no need. Plant normally require few days only. So it is also easily observable. So the characteristic of this plants is easily to observe. It means that when we compare, make a comparison like tall and short. So we can directly compare. Also, this plant is tall, this plant is short. When you compare the flower, also this flower for pea plant flower either is purple color or white color. Purple color or white color only, right? We compare the length of the plant, either it is tall, either it is short only. When you compare the wood, the seed of this pea plant, either it is yellow or green color like that. So it is very easily to observe. Don't need to make a and don't need to do the more additional analytic lah. Meaning, and then the fourth reason is it can be self or cross pollinated. Yeah, but normally many plants can be like that lah. So it either can self pollination and cross pollination. And then the lastly is available in many varieties. So it have many types of varieties. So this about the variation lah. I think you guys already know what is meaning of variation, meaning that it can be many types of trait lah. Got plant is tall, got plant is short, got plant with flower, with purple flower, got plant with white flower, like something like that. So that is why that they choose the he choose the pea plants as his experiment. So before I proceed to the his result, the experiment results, you have to know these all terms first in inheritance. So these are all the important terms that you have to know. Actually, this term you already study in SPM. SPM. And where we did. Okay. Actually, this term you are already learned in SPM. So I hope that everyone still remember the meaning. So normally many people confused about characteristic and trait. Yeah. But so actually characteristic is the observable feature of an organism. For example, like human length. What is your length? Itu panjang ketinggian awak, ketinggian awak. Sama ada awak adalah panjang atau pendek, macam tu. And then trait is the variant of the characteristic. So when we use ketinggian, sama ada awak adalah sangat tinggi atau awak hanya sangat pendek. So tinggi dan pendek itu is the trait. So if we use the pea plant as example, flower color. So flower color is characteristic. So what are the flower color? Either it is only purple color or it is only white color. Right? Purple color or white color. So purple color and white color is the trait. Another example, when we compare to the length of the flower, length of the plant, either it is very tall or very short, right? So tall and short are the traits. The length of the plant is the characteristic. Okay, so I hope that you guys know about this. So you guys have to know lah. And then trait, and then gene. I think gene, I don't need to explain. Right. All of you should know what is the meaning of gene. So it is the segment of DNA that serves a unit of heredity. So it contains genetic information. All of our information actually is come from gene. And then earlier is the alternative version of the gene. You can say that it is the segment of the gene. You can say like that. It is the segment of the gene. And then genotype and phenotype is also famous. Genotype is the genetic constitution of organism. So it is the gene content within us, within our cells. Phenotype is the physical appearance organism, like flower. It appears in like pea flower plant. It appears in purple color, but actually is it meaning that the gene the gene of this plant, all of the gene in this plant is totally purple, only contain purple color gene? So nobody knows. So not meaning that when the pea plant has purple color flower, not meaning that that plant does not totally have the uh white gene one. You have to know that. Not say that when the plant is or have the purple flower color, or then this plant is totally only have the purple flower gene only. No, not say like that. So we have to differentiate between these. So genotype is the gene information, phenotype is the physical appearance. Homozygous and heterozygous, I think this one you also know it. Homozygous having two identical alleles for a gene, heterozygous having a different alleles for a gene. So normally when we talk about the gene and allele, normally we will have the one gene normally have two allele. So one gene have two allele. Normally in Mendel that say one gene normally have two alleles. Homozygous mean that two identical alleles for a gene, meaning that this gene contain two identical alleles. So like either it is baby capital A or smaller a only. So actually capital A meaning it is dominant, and then smaller a is recessive. Okay, so dominant, what is mean by dominant? Dominant is the earlier can be expressed in homozygous or heterozygous. So heterozygous meaning it is the hybrid plant, meaning that the gene of this organism, it contains the hybrid genetic one. So it contains the dominant gene and also recessive gene. So it have both. But dominant characteristic, if that say we say that the uh, plant is dominant, meaning that the earlier expressed in either homozygous or heterozygous, meaning that this plant will express the dominant characteristic only. The recessive characteristic is not will be expressed like flower lah. So and normally the purple flower is dominant, white flower is recessive. So if it is the heterozygous one, so it will only produce purple flower. The white flower, yeah, it contains white flower gene, but because of it contains one dominant allele at here, so it will only express purple flower one. So the recessive gene is not expressed. So when we call recessive organism, so recessive organism normally only be expressed in homozygous only. So homozygous recessive only. So it only can be this only. AA. If we want to determine the recessive organism, it only can be one genotype only, AA. So this is we call as genotype. This symbol, all of this symbol is called genotype. Only on this genotype only can appear recessive characteristic. So like this, for this case, it will be white flower lah. So white flower will be the recessive. Okay. And then the P generation, F generation is the parental generation and filial generation. These are the when you have to use in schematic diagram. Okay. So what is the schematic diagram? I think you all have the memory about it. Normally it like that, right? So normally if you still remember like that, remember that. And then we fertilize, got this, inilah semantic diagram. Later I will explain it. So these are all the general terms about the inheritance you should know about it. Ini basic only, just a basic, very basic terms only. Okay, so I hope that everyone can understand. Okay, next is about the type of cross. So actually this is in the syllabus, but I think your school teacher not care about this. Never mind, we care. Okay, so type of cross. So this meaning that what is the cross meaning? Actually, it is the meaning when got one organism cross with food. But one organism that mating with food. So like this organism, any organism one, and then when their make it mates with who? Siapa? So this is the meaning of course lah. So we have 4 course we have to learn: test course, back course, reciprocal course, and self course. Okay, test course is the breeding of an organism of unknown genotype with a homozygous recessive individual to determine the unknown genotype. So this meaning that when we have the unknown genotype, let's say unknown genotype, I don't know this genotype, but we cross with the homozygous recessive. Ha, ini okay, a smaller one. So when we cross with it, then after that, we will get the result, right? The offspring result. So based on this offspring offspring characteristic, we can determine the genotype of this organism. So this is what is meant by test course. So when you see only when the organism is crossed with the homozygous recessive, then it is meaning that this organism is undergoes test course. Okay, next about back cross. Okay, back cross is breeding of a hybrid organism with one of its parents. So meaning when you have this organism, uh, you have this no, this one. If you have this organism, then when fertilized, then it will get this hybrid one. This is the parental generation. This is the F1 generation lah. Offspring. The offspring of these parents, ibu dia, apa? Ibu mother father lah. Okay, father. Okay, mother father. Okay, like that lah. Okay, when the father and mother breed together, they will produce the offspring with this genotype. Okay, and then when this genotype, this offspring, they back cross, meaning they are interbreed with salah satu ni. So it can be either ini or either ini. When it interbreeds either one of these, either it interbreed with his mother or father, when it interbreed the back toward from his mother or father, so from his parents so this meaning that it is the back cross okay. And then another one, reciprocal cross. Okay, so reciprocal cross actually is more complicated. Actually, it is same. Can say it is same. Looks same like a back cross. So it is a make cross between a pair of parents by using them in turns as female parent and male parent to obtain two reciprocal cross of A and B. So it intermediate change. So meaning when we reciprocal cross, meaning that if we do a cross, when the capital A genotype is mother, then recessive genotype is father. So reciprocal cross meaning we will proceed to another type of cross again, but this capital A genotype will become father. And then this recessive genotype will become mother. So dia terbalik lah. Dia buat cross tapi dia langsung terbalik. The sex of the organism is inverse. So version A and version B. So version A is when the mother is homozygous, and then the father is not homozygous dominant. Okay, and then the father is homozygous recessive. But the version B, they do reciprocal cross. The version B is the father become homozygous dominant, and then the mother become homozygous recessive. So they are tukar-tukar lah. It change the between a pair of parents lah. Okay, and then the self cross is very easy, meaning that they are cross dengan organism that have totally same genetic characteristic like this, like when it cross with the that totally have same characteristic or like this or like this, totally same one. Itu dia so macam dia totally same one, same genotype information, totally same. So this is the self cross. So I hope all of you know what are the difference between these four. Okay, so you have to know importance. So normally test course is about, because already say test course is when want to got what unknown genotype, then cross with the homozygous recessive, right? So when the test code is, we have to determine one of the important is we have to determine the genotype of this unknown species, unknown organism. So determine the genotype of organism and the express dominant phenotype. We have to know which phenotype genotype is express dominant one. Either it is tall or short. Either it is because at the beginning, George Mendel, they know which tall or short is dominant. Dia tak tahu ni. Dia buat curse ni, dia baru tahu. Oh, tall is dominant, short is recessive. Dia baru tahu pun. So, also able to differentiate between homozygous dominant and heterozygous genotype. And then identify the presence of hidden recessive earlier in individual with dominant genotype. Okay, then about is backcross. So backcross is we introduce the genotype of one parent into the genetic mapping or makeup of its offspring. So they want to maintain the genetic of the parents and then the preserve and enhance the desirable trait from a particular prevent in subsequent generation. So it have to maintain the also maintain the genetic information to enhance the desirable trait. The scientists don't want say that when they when they do a random cross or not do the back cross, the parents genotype information will be disappeared. So they do not, the scientists don't want that because they found that that kind of characteristic is very useful to human. So when that characteristic is hilang, disappear in the world, so it is not the human will lost will be lost from the knowledge and advantage from that organism. So they have to maintain and enhance that desirable traits. So it is very useful in breeding program to fix specific trait in a population. So they need to fix the trait maintain the traits in that kind of population. They don't want to have the imbalanced trait Now, don't want to say that one population in original that population has tall and short one, it is balanced. But suddenly they only found that all of the plants only have all of the plant is very tall and then the short plant is very few only. They don't want that. They have to balance the traits in that kind of population. And also used to obtain the pure lines of organism, because they normally the scientists if they want to have a pure organism, especially when you want to have a dog and cat, right? Dog and cat have also a different type of genotype, right? Normally some people is more focused on pure one. They have to a pure organism one. So when you have to one pure organism, so one progress, one of the procedure you can obtain is using this black cross to to make sure that the offspring produced is also have the pure organism, which is not hybrid one. Either it is like that or like that. It is pure one, not this. Ini is hybrid. Ini dia tak mau. Kami nak pure punya. Okay. And then reciprocal cross, okay, this one is normally is to investigate between the sex lah. So investigate the influence of maternal and paternal sources of genetic material on the trait. So when using this cross, although the genotype are same, but they have to investigate, make a research whether the sex the difference of sex got the effect on the offspring. Although the genotype are the same, or totally same, but maybe there are some genetic material in the sex gene got will affect that kind of phenotype on that. So they have to investigate. So and also help to discern whether certain traits are influenced by the genotype of mother or father. Yeah, because sometimes maybe in when using mother, yeah, it follow, but when using father, not follow. Why not follow? Because there are some certain traits on inside the mother or father got something meaning very very fine information about this to influence this. So the scientists need to found that kind of information. So that is why they do reciprocal cross to prove this result. And then the provide insight into the role of parental specific genetic factor in the inheritance. So also lah, they need to have to know what are the role of the mother and father genetic can feel and can influence, can affect the inheritance theory one. Okay. And then self-cause. So normally self-cause is the examine the inheritance pattern within a single organism. So this one is just want to know the inheritance pattern. So meaning that they have to know lah, or for this organism, the inheritance pattern is like this. Because actually, if why we have to differentiate between Mendelian and non-Mendelian, because not all type of the organism follow the Mendelian pattern one. No. So actually, if they have to examine the inheritance pattern, they need to do the experiment. Normally, the scientists will do the experiment to examine this. So normally, they will use self-cause because self-cause is very easy to determine compared to the other types of cause. So normally, self-cause is normally using heterozygous one. They will not use homozygous because homozygous directly your result is totally homozygous one. But heterozygous, the result is very complicated. So that is why we have the law of independent assortment. So because of this, we can know about the inheritance pattern for the pea plants. Okay, used for studying traits that are controlled by recessive alleles, reveal whether the organism is homozygous or heterozygous. Okay, so that is all about the cross. Okay, next is about type of breeding. Also there are 4 types of breeding you have to know. Actually, breeding and cross is similar. Normally is similar, but breeding is more about the offspring. Normally breeding is more about in the offspring one. So pure breeding, when we say pure, meaning that all of these offspring carry the same genotype trait as the parents by crossing with 2 homozygous individuals. Okay, so meaning that when this offspring contain this genotype, okay, so it will breed with uh with the with the individual that totally have this genotype one. So either it can be parents or it can be other type of organism. Not only limited in parents only, but normally we find parents because parents is more easily when we compare to other type of organism. But we can use other types of organism that has similar characteristic with this original organism, then they breed together to produce the offspring. Okay. Okay, next is selective breeding. Okay, when we have the selective, meaning that is human choose one. So human want to choose which type of the characteristic we want. Because human is very we can say it is very need to have a useful trait one. If that organism does not have useful trait, human don't want one. Don't want this trait one. If that trait is very useful, so human want. So normally in the most example is the pet lah. Kucing kan. So kucing pun ada salah satu kucing kulit dia sangat cantik. Semua sangat cantik kan, semua orang suka yang cantik punya kucing, kucing atau itu cantik punya kucing. So so want to breed with the kucing yang sangat cantik, normally they will use the selective breeding lah. So this one can ensure that itu the cat that we produce will be very we have the desired characteristic we want. Kami nak cantik punya. So kami pilih lah, ibu bapa dia memang cantik punya, memang handsome punya kucing, then they breed together. Normally the offspring they produce is also very cantik or handsome punya. They use like this lah. So if you normally like when people I want nak itu anak yang sangat cantik, atau awak nak anak yang sangat handsome, itu oppa, atau K-pop punya oppa ke, carilah awak yang sangat handsome juga. Tapi awak sendiri pun kena cantik juga lah kan. Kan awak sendiri pun kena cantik. So if you are cantik, then awak cari orang yang sangat handsome. So this is the selective breeding lah. So meaning that anak yang anda lahirkan ada kebarangkalian tinggi untuk anaknya awak adalah cantik atau handsome jugalah. So this is because selective breeding. So we need to select one. Kami select punya. Bukan voluntary punya. Bukan saya rasa ini satu boy okey lah, saya make with dengan awak lah. Bukan macam tu. So normally this one if we apply in plant, so normally apply in plant, so normally in vegetable lah, in foods, when we make together, then we produce the desired characteristic. So outbreeding and inbreeding. So outbreeding is when the individual met with the individual that totally does not have the common genotype characteristic. So normally if you see this example, so normally when we have the it has the offspring like that, right? When capital A and smaller a interbreed together, they will produce this offspring. So outbreeding meaning that it will interbreed with the organism that totally does not have the common genotype one. So it can be either that this or like this. Totally different one. And then inbreeding is it has the similar related to the genotype characteristic. So normally if this is outbreeding. And for inbreeding it will interbreed disease. Dia macam self-cross. You can say it is self-cross. Normally it is self-cross, but self-cross is among the same offspring. Because normally in plants, they only not produce one offspring only. They will produce many offspring one. Right. So normally if like that, if this example for this, it will produce five offspring like that. For self-cross is among these 5 offspring they cross together. But for this inbreeding, it not necessary to say that it must be crossed with the among adik-beradik mereka, saya nak cerita lah, the adik-beradik mereka. Self cross adalah untuk macam awak lah, as human as example, awak berkahwin dengan if I am man, saya berkahwin dengan adik perempuan saya atau kakak saya. Saya berkahwin dengan kakak saya. So if it mean that I am self-crossed, saya berkahwin dengan kakak saya kan untuk melahirkan anak. Senang cerita lah. But for this inbreeding, not meaning that saya kena kahwin dengan kakak saya. Saya pun boleh cari orang yang lain that have the similar genotype as me. So my genotype is is hybrid one, A and A, capital A and smaller a. So saya cari sama dengan genotype saya, dan saya breed dengan dia, kahwin dengan dia. So inbreeding lah. Meaning like that lah. If you confuse what is difference between inbreeding and the self-cross, you can see like that lah. It is not only fixed in with the same offspring, among the same offspring only. So it can be other organism one. Okay, so this is the type of breeding. So this is very general concept lah. I think this one you already learned in SPM 1 actually. Okay. So if you have any question, you just type in the chat box. I will always alert about this, about the chat box. Okay, nobody ask question. Okay, I will proceed. Okay, so now I will proceed to the result that George Mendel obtained. So actually, you guys, all of you know, Mendel it give two laws, right? One law is law of segregation, and another law is law of independent assortment. So in general, Mendel said that our chromosome, our gene, only have one allele. In Mendel lah, dia cakap, it's general lah. Nak cakap hanya satu allele je. So this is we call allele lah. And then the entire is gene, right? So dia normally human got a pair of chromosome, but organism have a pair of chromosome, but so idea satu gene hanya satu allele je. Dia hanya satu allele je. So all of say that state that the characteristic of diploid organism are determined by a pair of allele. Ini maksud dia lah. So diploid organism like human lah, because that human is diploid organism, but diploid organism means that one actually got one pair of chromosome, but we have a pair of chromosome that totally is contain the similar genetic information that control the same characteristic. The difference is the trait may be different only. Maksudnya kalau ini gene dia control the flower color, it only control flower color only. Dia tak akan control seed punya shape, dia tak akan control juga the ketinggian pokok tu, dia pun tak ada control the shape punya V6 like that. Dia tak akan control ni. Dia hanya control flower color only. So this means that one gene, one characteristic, the pair of the characteristic, one characteristic is controlled or determined by this pair of alleles. A pair of alleles. Ini dia yang conclude punya law of segregation. Dia hanya control kami ada dua chromosome, a pair of chromosome, a pair of gene a pair of gene, and then this gene contain each gene contain alleles, and then these two alleles will control or determine control our characteristic. Okay, so this is about the conclusion he made that at that time about the law of segregation and we use until now. We can apply this until now. Okay, so how he proof? Because if you want to get the result like this, you need to have a proof. Not say sure only. Bukan cakap sure saja. Awak kena proofnya, bukti. Apa bukti awak? So in senang, saya either senang je. Actually, you already learned Stpm. I just repeat only. So when normally when you are imagine you are the George Mendel. Kalau awak nak proof ni, normally which kind of organism you can use. Normally if you want to prove. So normally I use the example is flower color. Okay, so flower color of the pea plant, either it is purple color, that is purple color, or white color. So purple color is capital P, which is dominant, and then white color is recessive. Okay, recessive. Okay, so now when the first time do the experiment, so normally the most easiest way is when we use ibu bapaknya, I think I hope you guys can differentiate between these capital P and smaller p. This one is dominant, this one is recessive. I don't want to say dominant, purple color, purple, and then this one is white. Okay, ini P generation. Ini saya saya tukis schematic diagram. This is what we call schematic diagram. Okay, when we close with this, okay, next when we have to make lahirkan anak, apa yang kami perlu? Gamete, right? We have to have gamete. We need to produce gamete. Right, we have to produce gamete. Same as plant, we have to produce gamete. So how to produce gamete? So we have the process we call meiosis. Wrong spelling. Okay, when we have meiosis process, so this genotype, this organism, purple flower organism, it will produce the one type of gamete only. That is purple flower punya gamete. It will only produce the gamete they produce purple flower only. Purple flower gene punya sahaja. Purple flower early only. So this is gamete. So for this, it will only produce white color punya earlier only lah, because it only have white earlier, one type of earlier only. Both earlier is only or is white, so automatically it will become white. Okay, then we have fertilization, right? So when we make a fertilization, so it will produce F1. So the offspring that will produce of among this ibu bapa ni, it will produce this one capital P and smaller p, but the phenotype of this plant is purple color. Purple color one. The flower color for their offspring is totally purple color. So maksudnya kalau dia melahirkan 100 anak, 1000 ke 10,000 million pun, dia akan semua flower color dia adalah purple sahaja. Tak akan ada lain color dia. So Mendel pun pelik. Why like that lah? Mendel pun pelik. Kenapa saya buat banyak eksperimen tapi semua purple je. Tak ada white punya. Tapi ada white kan. Kenapa semua purple? Mendel pun pelik. Saya pun pelik. Ada sesiapa tahu? Maybe you don't know. I don't know. But actually, I think you know because you already learned. Okay. So if like that, Mendel need to know the inheritance pattern of this plant, of this flower color one. So what he do is it will interbreed with another. So it will interbreed with self-cross, or it can be interbreed with test cross first. Okay, we interbreed with test cross first. Okay, test cross first. Smaller p. Okay, test cross. Because why we need to interpret test cross? Because we have to determine the dominant characteristic or recessive trait, dominant trait and recessive trait. Because at that time Mendel pun tak tahu. Dia pun maybe dia tahu juga lah, or maybe purple is the dominant one lah, but it have to prove. So normally if you do same, it will undergo meiosis also. But when it undergo meiosis, it will produce two type of gamete lah. It can be either P, capital P or smaller p. Capital P is purple punya gamete or white punya gamete. So meiosis and then gamete. Okay, and then this one only one gamete lah. Hanya satu gamete je lah. Okay, when they do fertilization, do both of these, when fertilization, they found that they will get two type of offspring. So this is F2 lah. Okay, they will get two type of offspring. So sama ada anak dia boleh jadi purple. Dan juga anak dia boleh jadi white. Maksudnya apabila mereka buat this cross, this type of breeding, the Mendel found that they can either get got two kelompok lah. Satu kelompok flower purple punya, satu kelompok white punya. Oh, it is very interesting because it is very interesting. So from this, actually Mendel pun boleh tahu lah. From this, the phenotypic ratio you get is 1 to 1. 1 to 1. 1 purple, 1 white. So that is why we had the phenotypic ratio 1 to 1. I think all of you know law of segregation, either you can get 1 to 1 ratio or 3 to 1 ratio. So this is one of the phenotypic ratio you get 1 to 1. So this is about the test course when you use test course with it. So from this aspect, from this, it also can be proved that purple flower is dominant, white color is recessive. From this, they will know already. Because they found that when the purple flower punya hybrid cross together with white color, so dia akan keluarkan anak yang juga ada purple dan juga white punya. Kan? Kenapa? Kerana ini ada meaning that this organism ada this hybrid organism ada white color punya gene, tapi dia tak ada express saja. It does not express only because compared to here, because these two organisms, when you just see it, actually the mating is almost same, is the characteristic is same, because also purple color made with white color, but this result you get the result all are purple, but this you will get the result one purple one pink, one purple one white punya. So ada dua result berbeza lah kan, although the characteristic almost the same, is similar, but it get two type of result. Why? Because of the differences between genotype. One of the genotype is totally pure, one genotype is is hybrid one. So that is why you will get the ratio about 1 to 1. And then from this, we also can know purple color is dominant and white color is recessive. So this one is to determine the dominant trait and recessive trait only. So he used the test course to make a evidence. Okay, now we have to know the inheritance pattern of this plant. Okay, so normally how do they do is they will use the self-cause of hybrid. So meaning that they will use ini punya. So I use different color. Dia akan guna yang ni, cantum guna yang ni, campur dengan sama saja. Totally sama. Adik beradik mereka. Kerana yang ni dia akan menghasilkan banyak adik kan, adik beradik kan. So dia akan campur sama je. Okay. So same as it will produce gamete. So this one they will produce four gametes lah. This one will produce two type of gamete. This one also will produce two type of gamete. So when they do fertilization, it will found that this one is okay. They will found that they have the 4 possibility of genotype, right? But for the phenotype, if you compare the phenotype, all of these 3 is purple color, and then only one is white color. So meaning that there are 3 over 4 probability to get purple color. So maksudnya kalau ada lahirkan 4 anak, 3 anak adalah purple color punya. Hanya 1 anak is white color. When they do the experiment. So that is why that the phenotypic ratio for this, so the ratio for this plant is 3 to 1. So all together have 4 types of offspring, and but 3 are purple color, and only 1 is offspring. So when we use genotypic ratio, it will give 1 to 1. 1 to 1 is one is and for one is homozygous dominant, is pure, and another one is homozygous recessive, is white color, and two is the hybrid organism. So that is why the ratio is 3:1. And from here, we also know that white color totally is recessive, because white color appear very little. When you cross it, white color is appear very little one. Only purple color sahaja. Kebanyakan awak tengok semua purple color. White color hanya sebahagian sahaja. Right. So that is why when they do this kind of experiment, they will conclude that the characteristic is controlled by a pair of the earliest. So based on these two earlier, it can give the characteristic lah. Ini schematic diagram is when they do the experiment, the George Mendel buat banyak eksperimen, dia concludekan berdasarkan schematic diagram ini. Dia dah buat banyak eksperimen, it will give the ratio. They will already know the final ratio one, because based on the experiment they make, they already have the final ratio. Just that they use the schematic diagram to prove this ratio is correct. Okay, so these are all about the law of segregation. So ini senang je, very senang lah, I think. So I hope no question about this. Got any question or not? Okay, no. No question. So semua sangat pandai kan? Very smart people. So okay, fine. Okay, next law is about law of independent assortment. Okay, so this law is about it involves two characteristics already. Just now, it only involved one type of characteristic only. But this one, it involves two characteristics. How about when we compare two characteristics? Because that one is only one characteristic. Now we compare to two characteristics, two different characteristics. So as you know, as the law states that, law of independent assortment state that each pair of earliest in a gene can combine randomly with either of another of earliest for different genes. So meaning that when you have the two characteristics, meaning there are four earliest already lah. So these four earliest, each pair of these two pair, got two pair, right? Each pair of the earlier, they can combine randomly punya. Dia akan combine punya. Crossing over, right? We have crossing over, right? So crossing over theory also comes from this inheritance theory. So that crossing over, right? So state that each pair of earlier in a gene, it can combine randomly with either of another earlier one. So dia boleh hybrid, dia boleh combine with this. It can combine with this one. For different genes lah, totally different gene punya. Yang ni same gene, dia tak akan combine lah. Yang ni totally different gene. So if you want to prove that, normally we will use this lah, this experiment. You see that this experiment. Okay, we use back color. Okay, we will use this experiment. Normally is we use the two characteristic is six color and also seed shape. So color is either B blind is either yellow or green. And then for the seed shape is round shape or finger shape. Okay, so if you see that yellow color is dominant, this yellow and round is dominant, green and finger is recessive. So this is we know already lah, we already know. And then they do the experiment to prove that law of independent assessment. I think this ratio all of you are very familiar with it, 9331. Okay, 9331. So how we get this ratio? So I think I need to give the example. Okay, so as we know that uh y when we make with when we know that it involves two characteristics, so we use the dominant and recessive first. So why we write got four letters? Because I already say first law already say Mendel first law of segregation already say one characteristic is control determined by two earliers, by a pair of earlier. So there are two pair of earlier lah. One pair. So there are two pair of earlier. So this one is dominant, totally dominant, and this one is totally recessive, pure linear. So normally, if you do the fertilization, this one you will get only one type of gamete. This one also, you only have one type of gamete only. YR and what capital YR and also small YR. Okay, and then this one, parental generation. This one actually you have to like full form. Meiosis, gamete, and then undergo fertilization. You will get you will get this one. F1. So you will get this hybrid offspring, right? So when you combine fertilize them, combine with them, you will get this offspring. So this one actually, if you see the outside surface, awak nampak je dia pun juga akan keluar yellow and round shape. But it already have the gene of the recessive gene already. Okay, so when this hybrid is self-close together, okay, when it's self-close together, sama-sama interpret together, it will get the gamete which is very totally different. Yeah, we will get the totally different gamete. It cannot just like that only. Bukan macam Mendel first law, oh this one you will get like that right, and then this one you will get like that lah. No, because law of independent assumption say that each pair of earlier it may be can combine with another earlier of a different gene. So meaning that this capital Y, it can be combined with this earlier. It also can be combined. It may be combined of this earlier also. Similar with when this Y, it may be combined this, it may be combined of this also. So there are the four possibility. So normally when we want to draw this schematic diagram, so actually you can also like that. You can also like that. But it is very complicated lah. So normally we use Punnett square lah. So we use Punnett square to more easily to see the result compare you use the traditional method. So when you use Punnett square, okay, so when you use Punnett square, so these are the trait of the gamete, which are the gamete that can be used. So firstly, we use this first for the upper part, and then this one is the left column, left 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 row. This one I use the first column one. So for this, it can be Y, it can be made with capital R. Okay, one column. This one, it also can be made with small r also. One column. Okay, next, next, next is maybe this small y, it can be made with capital R also. Next, this small y, it also can be made with I need to actually okay, this small capital Y, it also can be made with small r, right? And then this one, small y, it may be also made with capital R. And then this small y also can be made with small r also. So there are 4 possibility of gamete about this one. Only first one. So when we compare to another one, so actually sama aja kan. So when you compare YR, okay, then capital Y, small r. Okay, another one is small y, capital R. And then another one is small y, small r. Okay, so when we do fertilization, so this column, this bracket, empty space, this space will be made with this. So capital Y, capital Y, capital R, capital R. So this one you will get, I use different color. Okay, this one you will get this one. Okay, and then for this one you will get Y, capital Y, Y, and then one capital R, small r, Y, because this one capital R, small r. And then for this one, capital Y, one small y, and then two both capital R, both R are capital. And then this one capital Y, and then small y, and then capital R, and small r. Okay, next, for this one, both are capital Y, and then one capital R, small r. And then this one, both are also capital Y, and then both are small r. And then this one is one capital Y, and then one small y, and then one capital R, one small r. And then this one is capital Y, one small y, and then both R are small. Okay, then for this one capital Y also, but small y, one small y, but R are both capital. And then this one is one capital Y, and then small y, and then both are R is one capital and another R is small. And then this one, both Y are small, and then both R are capital. And then this one you will get both Y are small, one capital R and one small r. And then this one you will get one capital Y, one small y also. And you will get one capital R and one small r. And then this one you will get one capital Y, another one is one small y, and another one is both are small r. And then this one both small y and one capital R, small r. And then this one you will get all are small. Okay, so these are all the partners, are all of the possibility genotype that can be appear when when you breed with these two organisms, these two type of genetic same genotype organism. So there are total 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So there are 16 possibility of the genotype that can be produced. But normally we only see phenotype only. So phenotype. So we only say phenotype only. Okay. So first possibility we maybe can see there are yellow yellow punya yellow color and round shape punya. Okay. Kami kena kira. Apa yang yellow color and round shape, this mean that it will appear dominant characteristic one. Both are dominant one. Yellow color and round shape. Okay, so which one is yellow color? So we one by one check. Okay, so this one is yellow color, right? Betul. Round shape, betul. So this one got one already. One. Ini for this one. So saya akan guna pen biru. Okay. So this one I cancel. So this one yellow shape, yellow color, right? And round shape, right? Because R capital R, got capital R here. So meaning it also dominant one. So dominant characteristic. Okay. So this one, okay, got capital Y and also capital R. So meaning that it also have the dominant character. Both are dominant characteristic. So ini pun sama. Okay, for this one, same. Right, same right, because got capital Y and capital R, so meaning that it also appear dominant characteristic. So, okay, already have 4 already. Okay, so for this one, it also have capital Y, also have capital R. capital R. Okay, so we have also another characteristic. Okay, so now so far already have 5. Both are dominant now. Okay, for this one, is it okay? True. Yellow, yeah, it is dominant because yellow, but this R is not dominant because already does not contain a dominant, or both are recessive. So meaning that this one does not have the shape, the seed shape of this genotype is not round shape. So there are another possibility that is another better possibility is yellow color and wrinkle shape. So got one lah. Ada satu lah. Satu dekat sini lah. So, okay. Okay, for this one, for yellow color, round shape, right? Got 2 capital. So this one also, we can green color lah. Okay. 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 Next, this one. Okay. Y. So capital Y, small y. But so this one is yellow color. But now the shape is not the round shape already because both are recessive. So it is a wrinkle shape. So it is yellow color and wrinkle shape. So it is considered at here. Considered at here. Okay. Next one. Capital Y and then got capital R. So meaning that this is also yellow color and round shape. So it is dominant also. Okay, so now already 1, 2, 3, 4, 5, 6, 7. Already have 7. This one capital got capital Y and capital R. So meaning that this is also the dominant one. Right, dominant one, dominant yellow color and also round shape. So okay. This one, okay. Now Y. So small y, both y are small and R is capital. So meaning that this one, this phenotype is green color punya the seed color is green and then the seed shape is round shape. So this is another possibility. Another type of phenotype lah. So this is another type of phenotype. Okay. So this one is green color punya and then it give round shape. So got one at here. Okay. Tulis salah. Yang ni kan. Ayo, sorry. Tulis salah. Yang ni Y. Okay. For this, Y shape, green color, right? The green color because both are recessive and then this one is dominant. So green color and round shape. So this one is also considered here. Green color and round shape because got capital R, right? And then for this one, capital Y and capital R. So normally this one is yellow color and round shape. So no. Okay, yellow color and round shape. Okay, for this one. So this one is yellow color, but the shape is wrinkle shape. So yellow color wrinkle shape is here. Okay. Okay. This one is green color, but the shape is round shape. So green color round shape. So this one is considered as here. Okay. So finally, this one is totally small. All are small. So it is green color and also wrinkle shape. Right. So this one is another characteristic. It is green color and also wrinkle shape. So this one is totally is recessive lah. I can say it is recessive, right? It is totally recessive lah. So only one. So when you compare all, so this one got 1, 2, 3, 4, 5, 6, 7, 8. This one got 9. This 3, 3, 1. So the phenotypic ratio for this one is 9, 3, 3, 1. So how we get 9, 3, 3, 1? Because of this. So now we get the 9, 3, 3, 1 ratio. So how it comes the ratio for this ratio, how it's come. And then for this, okay. So hopefully all of you understand how to use Banach square, how to calculate the ratio 9331. Okay, so I hope that all of you understand. Yeah. So law of yeah. So because there are many names about this law, many names. So this is the very scientific name lah, scientific name about this. So there are another name is Mendel first law and Mendel second law. Mendel first law and second law. So about when we got about Mendel first law, is about law of segregation lah, and then it's ono monohybrid only. Monohybrid. Both monohybrid. And then another one is Mendel's second law, right? So for this lah. For Mendel's second law, the scientific name is law of independent assortment, but another name we call is dihybrid. Dihybrid inheritance and monohybrid inheritance. So monohybrid, when we say mono, meaning that it only involves one characteristic to prove Mendel's first law. And another one, dihybrid, dihybrid is it will involve two different characteristics to prove Mendel's second law punya. Okay, so I already proved all of you. So in conclusion, so I think no but me to do a conclusion. Okay, if it's I think a bit okay. So in conclusion, I do a conclusion. So Mendel punya first law, it about only involve one characteristic, and then the two ratio you will get is 3:1 or 1:1. So 3:1 ratio is when you do the hybrid self cross, self cross. 1:1 ratio is when you do test cross. Second law, you will also get the two ratio. So the most famous ratio is 9331, right? 9331. These are the most famous ratio. This one when you do the also self cross also. So when you do self cross, hybrid self cross. Another one you will get 1111 punya. Sama aja dia you will do the test cross punya. Yang ni punya. When you do the cross of this, you will get the ratio 1:1:1. But this one I not sure. Actually dia sama aja dengan ni, tapi dia tak perlu buat Banach square. You directly can do already. So you will get the 1:1:1 ratio. 1:1:1 meaning that there are four possibility lah. Also have four possibility also. This one you will get 9331. Okay, so these are all the ratio that you will need to learn, you need to know for Mendelian punya. Ini Mendelian punya. You have to know it about the theory. Why? Because when you proceed to non-Mendelian, all of the ratio that obtained in non-Mendelian also is according to the Mendelian one. Because the non-Mendelian concept is the further scientists they do already. When the further scientists they found that they also use the Mendelian punya concept, but they found that the result obtained is totally different from Mendelian one. But they just modified it. So normally in examination, they will need you to compare between Mendel and non-Mendel one. They will give the one situation and then you need to explain is it follow Mendel or non-Mendel. So if you don't know the Mendel punya, the basic one, so when you proceed to non-Mendelian punya, you will be very confused. Okay. So please alert, although Mendel is very easy, I can say it very easy lah. Memang sangat senang je. So senang senang sampai semua tak nak belajar because you already learn in SPM. You will think that ayah mental dah sudah belajar lah, tak perlu belajar lagi lah. Kita belajar apa kamu? Saya nak challenge saya terus belajar 6 Mendel punya. Fine. If you think you are very expert, you can skip it. Fine. But if you're not expert at all, please revise back again Mendelian inheritance. I already explained it, so hopefully this you can know about what talking about for Mendel. Okay, so I hope everyone can can ingatkan memory SPM kamu. Memory SPM dah letak otak belakang kan. So I hope that your memory SPM is in front of the brain now. Okay, so for actually Mendel dah tamat lah. Okay, so actually I don't know you guys want me to proceed non-Mendelian or not, because non-Mendelian is very long topic and I see now the view is only 8 views, so I don't know you guys want me to proceed non-Mendelian or not, because non-Mendelian I will separate into 3 parts. My slide lah, I will split into 3 parts. One, but if you guys want me to proceed non-Mendelian, I will only sentuh first part only lah. The second part and third part will be tomorrow lah, tomorrow session. But if you don't want proceed now, so maybe we'll proceed tomorrow lah. So I ask your opinion about you guys. Want me to proceed non-Mendelian or not? Today I proceed. I only proceed first part only. Second and third part I will proceed tomorrow. Cepat. Okay, Duha, he hope me to proceed. Others, only one, because only 10 views. So others. Okay, so got 2 people. So okay. So more 3 people say need to proceed. Okay, so I will proceed. So yeah. Okay. Non-Mendelian, yeah, because non-Mendelian is the part that most of you are very confused. Because when you compare with Mendelian, yeah, memang punya. So, okay, non-Mendelian my slide is divided into 3 parts. So for today, I only will proceed to part 1. So part 1, what you will learn is about these 4 only. Incomplete dominance, codominance, multiple earliest, and letter gene only. Second part is about link gene, and third part is polygene. This one is first, second, and then third part. So polygene epitasis pre-degree analysis. So this second and third I will continue tomorrow. For today, I will only proceed for these 4 only. Okay. So firstly is about incomplete dominance. Okay. Saya cakap cerita je. I give you a story. Because if you see the form is very, maybe you don't know. Saya cakap story je. In Mendel, when you recap for about the Mendel one, when your Mendel, you recap Mendel cakap we only have 2 types of earliest, a pair of earliest. Either it is dominant or recessive, and then the characteristic we appear for Mendel. Mendel dah cakap it only have one dominant, 2 earliest, right? two alleles. One gene is controlled by a pair of alleles and this allele either it is dominant or recessive only. Only. And then the phenotype that we appear either it express dominant characteristic or express recessive characteristic only. The phenotype. Dia hanya either dia appear dominant atau recessive. Dia tak ada tengah-tengah punya. Mendel dia cakap, tapi betul ke? Betul ke? Semua organism apply ni ke? Betul ah? Kan? First, then you say that one characteristic is only controlled by a pair of allele. Not all. Bukan. Bukan. And you say that one characteristic is only controlled by two alleles. No. No. And then you say that alleles only have one dominant and one recessive. No. Because some have two dominant one. And then you say that phenotype will only express the dominant one. Either it is dominant or hybrid. Hybrid will also express dominant characteristic. And then the recessive or recessive only. So, but no. Siapa cakap? Dia cakap je, Mendel cakap je. Tapi in real situation, in the real world, is it all organisms follow this? No. The answer is no. So that is why we have non-Mendelian inheritance. So these are all the scientists when Mendel they publish this research, this result to all around the world, and then the all around the world of the scientists they make a modification, they modify, and then they found that not all type of organism will totally follow this Mendel punya. Bukan, tak semua. Tak semua. But normally the theory is same. Just that there are some modification some modification. So that is why we will have many types of these non-Mendelian inheritance. Okay, so this first type Mendelian inheritance is incomplete dominance. So what is meant by incomplete dominance is the allele that is not completely dominant and F generation have phenotype somewhere between the two varieties of two parents is called incomplete dominance. Meaning that if you follow the Mendel punya use pea plant pea plant flower flower punya pea plant flower punya sorry. Ikut Mendel, kalau kami ikut Mendel lah. Kalau ikut Mendel kan dia cakap macam dia cakap kalau offspring dia kalau like this or like this. Kalau lah ada tiga ni kan like this. This one maybe you say that the dominant characteristic is tall. Okay, tall. So this one pun tall juga. Yang ni short lah. Kan? Ini Mendel cakap. Tapi in incomplete dominance bukan, tak betul. Incomplete dominance yang ini hybrid that is not completely dominant, when we call that the completely dominant, and then the generation, the phenotype is somewhere between the dia tengah-tengah punya, dekat tengah-tengah punya. So this one will totally have the different characteristic. Dia tengah punya, at the middle one. Middle one. So somewhere else between these two varieties of parent punya. So parent dia, ibu dia panjang, dia sangat tinggi. Bapa dia sangat rendah. Tapi anak dia ada satu kebangkalan anak dia macam tengah-tengah je. So in example, one of the example is this dragonfly. Dragonfly flower. So if you see here, dragonfly is dragonfly flower, right? Not dragonfly, snapdragon, sorry. Snapdragon flower. Snapdragon flower is one of the famous example we use. So if you see here, we use this red color flower and also white color flower. So kalau awak ikut Mendel, maksudnya dia akan akan ada merah je atau putih je. Either it is red or white only. But in this flower, it's very special. When they made together, it will give pink color one. Wow, fantastic. So meaning that it only have one kebangkalian, one type. Meaning that when red snapdragon and also white snapdragon, they made together, they will give the pink color one. Totally different punya. Very totally different because it is hybrid. If you see here, it will give the genotype of CR and CW. So this genotype will give the appear pink color one. It will not appear red color or even white color one. Totally different. It will only appear pink color one. So, and then when they do the self-cross for this pink color, they will get the result of this. So you will get the result one red color, two pink color, and one white color. Actually, kalau ikut Mendel, if you use Mendel punya, you will get the result 3-1, right? Mendel punya 3 is dominant and 1 is recessive, right? Ikut Mendel. Tapi if you ikut ni incomplete dominant, sorry, it will not ikut Mendel. It will give you 1-to-1 ratio. One red, two white, and one pink. Sorry. Two pink, one white. Sorry. One red, one white, and two pink. One red, two pink, one white. So it will give you this different ratio one. Dia memang tak ada ikut Mendel. Why it not ikut? Because the hybrid does not express the dominant trait. It will give the trait that is somewhere else between parents one. So merah campur putih, jadi pink color lah. Jadi pink color. So this ratio is 1-to-1. So I hope understand about this. Okay. So no question? If you have any question, you just type on the chat box. I will. Ayo, slide still in conclusion. Oh, sorry. Ayah, saya cakap banyak kali lah. Alah, aloi. Alah. Baik. Very sorry. Aloi. Why like that? But already. Ya, saya cakap. Dah nampak? Yeah, very sorry. But I think the introduction part, I think I not write anything. I think you guys can still hear that. So I just recap so non-Mendelian inheritance is like this. So first part I only cover these four types only. Okay, now I already proceed to incomplete punya. So actually lah, saya berbezakan dengan Mendel dan non-Mendel. So incomplete Mendel punya, if you ikut Mendel, actually you will get these two result, right? Okay. I hope that everyone can hear again. Okay, so if you ikut Mendel punya Mendel, so you will get the result. You will get the result either is this one, hybrid one, or recessive one, right? So if you got these three type of genotype, all of the phenotype is maybe this one is tall, this one is tall, so only this is short, right? If you compare, you will use Mendel. But incomplete dominance is very different. That the earlier is not completely dominant, meaning that this is hybrid, but it is not completely dominant. And then the F generation offspring there have the phenotype somewhere between the varieties of two parent punya. So dia dekat tengah-tengah punya. So kalau tengah-tengah, middle punya. So this is totally not follow the Mendel lah. Karena Mendel hanya cakap phenotype either is dominant or recessive only. Ini Mendel cakap. But non-Mendel in real situation not say that all of the organism follow that. So it will have the some what kind of the sometime ada middle part punya. Middle punya. So somewhere between the varieties lah. So the example of the incomplete dominant is about uh the example of the incomplete dominant is snapdragon flower. Snapdragon plant. Snapdragon punya plant punya flower color. So snapdragon either it can be red color or white color. But if you according to the Mendel, Mendel punya, you will see that you will see that it will appear pink color punya, or one pink color punya. Maksudnya tak ada dominant, tak ada recessive lah. No, so in this case there is no dominant and recessive. We cannot say that red color is dominant, white color is recessive. Cannot. Yeah, so all of them we can say they are same lah, parallel 的。So snapdragon pink color. So when they do a cross between them, it will get only the one type of offspring. It is pink color flower. Pink color flower. So it will get the genotype of this. So this genotype will give the pink color punya flower. And then after that, when they do the self-cross again between the pink color flower, they will get the ratio about 1 to 1. So one red color, two pink color, and also one white color. So if you ikut Mendel punya, so Mendel actually it will give you 3 to 1 ratio. 3 is about dominant. If you ikut Mendel, maybe lah, red color is dominant, white color is recessive. You will get 3 red color and 1 white color, or ikut Mendel. But unfortunately, in real situation, the snapdragon plant, the flower of color will not ikut this. It will ikut ni. 1 to 1 ratio. So 1 red color, 1 white color, and also 2 pink color punya. So it is totally different from the Mendel one. I just repeat only. So sorry of that. Okay, I think the slide is already. I think need to. Okay. Okay. So now I will proceed to core dominance. So actually, in conclusion, for incomplete dominance, meaning that dia ada satu tengah partnya. The main difference is about hybrid organism. Hybrid punya organism. Kalau hybrid organism punya, ikut Mendel, dia akan express dominant phenotype characteristic. But unfortunately, incomplete dominance, for the hybrid organism, it will express the variety somewhere else between parent punya. So white, merah dan putih tu somewhere else macam pink color lah. Macam pink color lah. So dia langsung tak sama dengan Mendel. Okay, core dominance. Okay, so for core dominance, it is the heterozygous individual that have both pair of allele are equally dominant and expressed in the phenotype in equal magnitude. This one is meaning that if you ingat, still remember Mendel, dia finalize allele must be on one dominant and one recessive only. But in real situation, there are some type of characteristic that contain two dominant one. There are two dominant one. So the mostly example is blood group and N blood group. Okay, so the most example is the N blood group. When you compare to the MM blood group, so MM blood group, it does not contain recessive gene punya. It does not contain recessive allele punya. Dia ada dua allele, but these two allele both are dominant one. So that is why we call core dominance. because they are equally dominant. And then the phenotype they will appear equal punya. So bukan cakap blood M punya M punya MN punya ini MN punya hanya ada M punya or N punya. Dia dua-dua ada. Dua-dua ada punya. So if you do the schematic diagram tak here, when you do a schematic diagram, so blood group M and blood group N, when you combine it, it will get the hybrid one, right? Hybrid. So this punya phenotype is blood group. This punya it is blood group M N. Dua-dua ada punya. The phenotype dia ada dua-dua punya. So meaning that this kind of people, it have both punya phenotypic characteristic. M pun ada, N pun ada. Okay, and then they do a self cross again to determine the inheritance pattern. When they do the self cross or some other, okay, after that they will get the result like this. So these are the result. So it will get the result 1, 2, 1. 1 is M, 2 is NN, and another one is N. So it has the same ratio with the incomplete dominance one. Sama ada. Dia ada sama ratio with the incomplete dominance. So, dia tak sama dengan Mendel apa? Mendel cakap hanya ada earlier, it must have one dominant and one recessive. But for codominant process, meaning that both are dominant. It will contain more than one dominant punya. It does not necessarily only have one dominant. Actually got two dominant. So that is why we call codominant. So for this codominant, the ratio you will get is 1:1, same as incomplete dominance one. And another example is about ABO blood group. ABO blood group is also one of the example. For this codominance, but the ratio is not you will not get 1:1 ratio lah, because ABO blood group is for this one multiple earliest, because the phenomenon when there are more than two alternate form of particular gene that occupy a gene locus. So sama if you compare to the Mendel, so Mendel cakap one type of gene only have a pair of allele only, controlled by a pair of allele, and only either one dominant and one recessive. Tak akan ada more than two allele one. Ini Mendel cakap. But in multiple allele punya case, it will have more than two. So the most example is blood ABO lah. ABO blood group. Why? Because ABO blood group got three allele. Either it is allele for A, B, and also O. So it already have three types of allele already. But finally, only a pair of allele control one characteristic. It does not mean that these three allele boleh campur together. Tak boleh campur punya. Tak ada campur punya. Hanya either you will get these or these. Also got this got this, or either got this. So got 5 probabilities lah. So if you compare to Mendel hanya 3 probabilities, and this one got 5 probabilities of the phenotype. Okay, so these are also totally different from the Mendel punya. Mendel cakap hanya 2 allele je, tapi ini cakap ada 3 allele. More than 2 lah, not only 3, maybe 4, 5, like that lah. But the most example is ABO blood group lah. And then ABO blood group also the example of the codominance. Why? Because A and B are dominant. Blood A and blood B are dominant, right? They are equally dominant one. So okay, so this is just the extract from the Campbell book. So earlier, so we have the earlier A and B, and also O, none. This one we use ANA, so all IO. Okay, so this is a blood group genotype and phenotype. So when the people that has the AB blood group, meaning that it will have the difference is it will have the A punya antigen and also punya B punya antigen punya. Dua-dua pun ada. Red blood cell is a fake carbohydrate, so maksudnya the erythrocyte of the erythrocyte surface of this erythrocyte it contain A punya and also B punya. So dua lah. So this is why we call as the multiple earlier and of the multiple codominance. For multiple earlier because of it has three different earlier at all. Okay. So these are the meaning of the multiple allele. Okay, for another one is lecter gene. So lecter gene is that if phenotypic display of certain gene, the people will die. Dia akan mati. Kalau ada orang, ada gene ni, dia terus mati. Tak boleh survive. So one of the example is the coat color of the mice. So normally the color of the mice either is yellow or gray only. Yellow or gray. But if it has the genotype of this dominant color, because yellow is dominant and then gray is recessive. Punya. So if you see the yellow color, so this one it can be survived because it hybrid. But if the mice have this genotype, capital Y, both are capital Y, so the mouse is die. They are directly die. They are doubly survive. Although it can survive, but maybe several months only lah. Maybe several months it will directly die already. It cannot survive with this gene. So for this part, it will have the ratio only 2:1. So it will produce yellow, and gray. So compared to Mendel is 3:1, right? 3:1. Kenapa 3? Because of this. Sepatutnya kalau dia tak ada mati, sepatutnya 3 kan. Tapi sekarang dia ada mati. It already died. So that is why we change to 2:1 only. Lecter gene meaning that there are one phenotypic ratio or one genotype, dia akan menyebabkan kematian punya, will cause death punya. And another is for human one is this lah, albinism disease. Although dia tak directly mati, but this if the people have this disease, normally it cannot survive very long lah. The lifespan of this human is very short. It's very short. So one of the two sisters, the other has mother. Most recessive homozygote are born to parents who are carrier for one phenotype. So if you compare to this, so when the parents they have normal phenotype and normal phenotype, but these two parents are carrier. They have the carrier this carrier. So dominant A is normal lah, normal people. This one recessive is the disease. This one carrier of this disease, albinism disease one. So kalau mereka interpret together, so they will get the 3:1 also get the 2:1 punya. So one is normal and another one is carrier, but one is albinism phenotype lah. Albinism lah. Why? Because and then normally people get this disease, they cannot survive. The lifespan is very short lah. So if you guys is interesting about this disease, maybe you guys want to become a doctor or not, you want to study this disease, you can study it by yourself. You can search about your Google. But normally, as I know, for these people with this disease, normally the lifespan is just very short lah. Okay, so this is also one type of example of lecter gene. Okay, so I think that's all for today. So all are four type of non-Mendelian, but incomplete dominance, codominance, multiple earlier, and also lecter. Okay, so if you have any question, you can ask now, and I will make a conclusion lah. So incomplete dominance, so normally totally non-Mendelian inheritance is totally different from the Mendel. You can make a comparison between the ratio of the Mendel one. So incomplete dominance is a digit, is a part that the by offspring that has the ability between somewhere between parent punya. So dia akan tegak tengah-tengah punya. So one of the example snapdragon lah. So this one is more of hybrid punya ratio. So hybrid punya. Kerana hybrid of this organism, dia hybrid, dia tak ada express phenotype ratio punya. Dia akan express totally another trait. So normally this one incomplete dominant, you will get the ratio 1:1. You need to remember it. Codominance is the organism that they have two dominant. Compared to Mendel, Mendel cakap hanya satu dominant, satu recessive, but codominant have two dominant equally. So normally, one of the example is Bloodgood, the ratio is also same 1:1. And another example is ABO, but ABO does not follow this ratio because ABO also the multiple allele. Multiple allele does not have fixed ratio because multiple allele, it may be 3 allele, 4 allele, 5 allele. So one of the example is ABO blood group. It contains 3 allele, right? IA, IB, and IO. Why it also called the codominant? Because A and B are equal dominant. So that is why we have AB blood group. And then another one is lecter gene. So this one, if there are one type of the gene, either it is dominant, dominant, recessive, or hybrid, any type. If there are one type of the gene is appear at that organism, it will cause die. So if that die, so normally we cannot calculating ratio, we cannot include it lah. So normally the ratio is 2:1 lah. Compared to Mendel is 3:1, right? So it will give the ratio only 2:1. So I think that's all for this non-Mendelian inheritance. This is only part 1. Tomorrow I will explain all about all these non-Mendelian punya link gene, sex link gene, polygene, epitasis, and pre-degree analysis. So another 4 I will proceed tomorrow. So I think that's all for today. Okay, so got any question or not? So I think no question. So everyone very smart. Know what are the meaning of the incomplete Mendelian inheritance and non-Mendelian inheritance. Okay, so actually for now the class is end lah. Actually, so I first need to thanks all of you attend the class today. So if you guys have any questions, you can ask now. So okay. Okay. So for now, and I need to remind all of you this class does not have the recording lah, no recording at all. So if you guys want to buy the notes, you have to buy it. I already sent the link at there, right? Okay. So if you have any question, you just ask it. If not, okay. Okay, so if you guys want to buy the notes or video recording video, you can scan this QR to buy it. Okay, so if you guys know any question, you can leave your support now, and thank you for your attending. So for Indian, if you join, I wish you happy Deepavali. And I don't know school holiday or not. If you got school holiday, happy holiday. If not, happy weekend. And we will proceed the class tomorrow. And maybe not change already time because today I have emergency issue for this night. So tomorrow will remain 8 o'clock. So we will proceed to non-Mendelian inheritance. So if you guys have any question now, can ask. If no, can leave now. So thanks for your joining. So if you want to buy the note or video, you can scan the QR code and fill in the information that you want. Okay, so thanks for watching. Thank you. Bye.